Hello everyone, welcome into the ITLS Academy. Myself, Dr. Himani, PhD in Tissue Engineering and Regenerative Medicines. And today we are going to discuss about the animal cell culture and its techniques. So, we are discussing about the animal cell. What is animal cell actually? Cell culture is the process by which prokaryotic, eukaryotic or plant cells are grown under controlled conditions, but in practice it refers to the culturing of cell derived from animal cell. So we can define it, animal cells are the basic unit of the life in organism. They are eukaryotic cells, meaning that they have a true nucleus and specialized structure called organelles that carry out different functions. Animal cells do not have plant specific organelles like cell wall which support the plant cells or chloroplast, the organelles that carry out photosynthesis. So in this picture we can see the structure of animal cell structure. All cells are surrounded by a cell membrane. You can see here, this is the cell membrane. All cells are surrounded by this membrane. This is called cell membrane. So the cell membrane is the boundary that separated the inside of the cell from the outside of the cell. The plasma membrane includes all the cell components which are suspended in a gel-like fluid called cytoplasm. This is the cytoplasm. You can see here. And the cytoplasm is the located of an organelles. So eukaryotic cells are distinguished from prokaryotic cells by the presence of defined nucleus. This is the nucleus. You can see here, and other membrane bound organelles such as mitochondria, Golgi vessels, Golgi apparatus, smooth endoplasmic reticulums, and chromatin, mitochondria. So many cells are present here. So prokaryotic cells do not have a defined nucleus. Instead, a region of the cytoplasm called the nucleotide hold the genetic material. This nucleotide or nucleus hold the genetic material. They also lack membrane bound organelles. Animals are the multicellular, meaning multiple cells work together to form the whole organism. In complex organisms such as humans, these cells can be highly specialized to perform different functions. As such, they often look and functions very differently from one another, even though they are all human cells. So you can see in this picture different common cells types, like this is the structure of stem cells, this is the structures of intestine cells, these structure of red blood cells, muscle cells, liver cells structures, and nerve cells. So complex animals such as humans have a variety of different cell types. Each look and functions very differently. So now we are going to discuss about the history of the animal cell culture. Although animal cell culture was first successfully undertaken by the Roche in 1970. It was the 1907. So it was not until the late 1940s to early 1950s that severe development occurred that made cells occur widely available as tools for scientists. First, there was the development of antibiotics that made it easier to avoid many of the contaminants problem that plagued early cells culture attempts. Second was The development of the techniques such as the use of trypsin to remove cells from culture vessels necessary to obtain continuously growing cell lines such as HeLa cells. And third one is these cells line scientists were able to develop standardized chemically defined culture media that made it far easier to grow cell. So now we are going to just discussing about the historical event in the development of cell culture. Cell culture arose in the 19th centuries when psychologist Sindra Ringer developed a salt solution that was able to sustain the beating of an animal heart outside the body. However, the principle of tissue culture was first evident in 1885 when Rocks stabilized the method known as tissue culture. You can see here in 1985, Rocks was the defined the method of cell culture. This one, and in 1910, the Carroll successfully cultured embryonic and adult tissue from a variety of mammals, and in 1912, Carroll successfully subcultured a cell line hundred of times from heart fragment of chick embryo. In 1940s, the use of antibiotic penicillin and streptomycin in culture medium decreased the problem of contamination in cell culture. So you can see the scenario of the development of cell culture. So in 1992, you can see the skin ethic product human tissue and neuron stem cell culture in vitro was developed, and 2002 the exploit tissue engineering was developed, and 2010 and now demonstrate 3D tissue and organs bioprinting techniques are growing on. So today cell culture is significant principle for research. In 2006, the US Congress divided a 1 million to various pharmaceuticals companies to develop cell cultured influenza vaccines, and there have been significant advances through the establishment of various centers that specialized and support cell culture research. Now, we should know about the cell culture. Actually, what is the cell culture? Cell culture refers to the process by which cells are grown in controlled artificial environment. Cell can be maintained in vitro outside of their original body by this process, which is quite simple compared to organ and tissue culture. So we can define of the animal cell culture is animal cell culture can define as the type of cell culture where the cell grow and multiply either in a solid or liquid medium as cell monolayer or cell suspension respectively to produce the primary cells. Then followed by subculturing of primary cell, it produces secondary cells to a defined cell number for the normal cells and identify for the continuous cells. So cell culture involves the distribution of cells in an artificial environment called in vitro, which is composed of the necessary nutrients, ideal temperature, gases, pH, and humidity to allow the cell to grow and proliferate. So Let's discuss about the in vivo cell culture. What is the in vivo cell culture? When the study involve living biological entities with the organism. And what is the in vitro cell culture? When the studies conduct using biological entities like cell or tissues that has been isolated from their natural biological environment. Example, tissues or cells isolated from the liver or kidney. So this is the in vitro cell culture method. And now why cell culture used for? This is the important things. Why we are using cell culture? First area where cell culture technology is currently playing a major role is model system for studying basic cell biology, biochemistry and physiology. Second one is interaction between disease causing agent and cells. Third one is effect of drug on cells. Fourth one is process and triggering of aging nutritional studies. And fifth one is toxicity testing study the effect of new drugs. And sixth one is cancer research study the function of various chemical, virus and radiation to convert normal culture cell to cancerous cell. So now we should know about the morphology of cell culture. Based on their appearance, cell in culture can be categorized into three main groups. First one is fibroblast-like. So what is the fibroblast cells? Fibroblast cells are the bipolar or multipolar, have elongated shape. You can see this is the bipolar or multipolar and elongated shape. You can see the shape of the fibroblast cells. This is the elongated shapes and grow attached to a substrate. This require a substrate for the growing. So this called is the fibroblast like cells. And second cell is the epithelial like cell. Epithelial like cells are polygonal in shape. You can see the shape of the epithelial cells. You can see this is the polygonal shape and with more regular dimensions and grow attached to a substrate in discrete patches. You can see here this is the discrete patches. See. So this is the epithelial like cell. And third one is the lymphoblast like cell. These lymphoblast like cells are spherical in shape. You can see in this picture structure of this lymphocyte spherical in the shape, usually grown in suspension without attaching to a surface. These type of cells not require any surface. So this is the lymphoblast like cells. Types of animal cell culture. On the basis of cell growth and division, the cell culture is two types. First one is primary cell culture and second one is the secondary cell culture. First, we will discuss about the primary cell culture. When cells are surgically removed from an organism and placed into a suitable culture environment, they will attach, divide and grow. This is called primary culture. So the cells can obtain directly by the mechanical method and indirectly by the enzymatic actions. Once the cells are obtained, they have to be cultured on a suitable container which is provided with all the nutrients required for the cell division and growth. The growth of primary either occur as adherent monolayer or on the solid medium or as suspension in the liquid medium. So let's discuss about the what is the adherent monolayer and what is the suspension layer. So suspension layer, these are the cells that do not adhere to the surface of the medium provided. Suspension cells are the types of cells that floated as suspension in the liquid medium. So, the property of the adherent cell include, first one is the suspension cells are anchorage independent. Second one is float as the suspension of cells in the liquid culture medium. Third one is the growth occur in the liquid nutrient medium. And fourth one is the suspension cells grow much faster than the adherent cells. And there is a short lag phase in the suspension cells. Enzyme action is not required for the dissociation of the cell. This is the main thing. Enzyme action is not required for the dissociation of the cells. Now we will discuss about the adherent cells. These cells adhere to the solid surface and produce a cell population in the monolayer pattern. Adherent cells sometimes refer as confluent cells, whether the cells merge or contract to fill the surface area. The properties of the adherent cells include, first is adherent cells are anchorage dependent. Second one, grow as a monolayer. Third one is growth occurs on a solid surface or we can say solid media. And fourth one is adherent cells fill the entire surface area of the container or vessel as a monolayer, and adherent cells follow the property of contract inhibitions. So whether the cell itself causes the growth of more cells to maintain the synchronized state by the chemical signaling, which does not permit the overgrowth once a monolayer is formed. An example of fibroblasts and epithelial cells are example of the adherent cells. So we have covered adherent and suspension cells. Now we are going to see the morphology of cell dependent on adherence properties. As anchor dependent, you can say like this is a adherent cells and this is the suspension cell. You can see the morphology of adherent cells must be cultured while the attached to a solid or semi-solid surface. Example is the MCF7 and PC3. This is the morphology of anchor dependent cells, and this is the morphology of suspension culture or anchorage independent. Can we grow floating in the culture medium? Can we grow the floating of the culture medium? Example K562. So difference between the adherent and suspension cell is adherent cells attach to a solid or semi-solid surface, but suspension cells grow floating in the culture medium. Second one, easy visual inspections under inverted microscope, but for the suspension cell you require daily cell count and viability determinations to follow growth pattern. And for the adherent cells are dissociate enzymatically like trypsin or mechanically, but for the suspension cell does not require enzymatic or mechanical dissociation. Growth is limited by surface area for the adherent cells and growth is limited by concentration of the cells. No agitations required, but required agitations in the suspension cells like shaking or streaming or the adequate gas exchange. For the adherent cells used for cytology, harvesting product continuously, and many research applications are required for the adherent cells, but for the suspension cell used for bulk protein productions, batch harvesting, and many research applications. Now we are going to discuss about the method for doing primary cell culture. Here are two basic methods for doing primary cell culture. First is the explant culture. Small pieces of tissue are attached to a glass or treated in a plastic culture vessels and bathed in culture medium. After a few days, individual cells will move from the tissue explant onto the culture vessel's surface or substrate, whether they will begin to divide and grow. The second more widely used method is speeds up the process by adding digestion or proteolytic enzyme such as trypsin or collagenase to the tissue fragments to dissolve the cement holding the cells together. You can see in this picture. This is the if we are taking the fish tissue, then minced or chopped by the Caesar, and digestion with the proteolytic enzyme will be started and placed into the culture medium or culture flask. So this create a suspension of a single cell that are then placed into the culture vessels containing culture medium and allow to grow divide. This method is called enzymatic dissociation. Now we will discuss about the secondary cell culture or cell line. When a primary culture is subcultured, it is known as secondary culture or cell line or subclone. The process involves removing the growth media and dissociating the adherent cells, usually enzymatically. So, subculturing of primary cell to different division lead to the generation of the cell line. During the passage, cells with the highest growth capability predominate, resulting in a degree of genotype and phenotype uniformity in the populations. However, as they are subcultured seriously, they become different from the original cells. Now, types of cell line. On the basis of the span of culture, the cell line are categorized into two types. First one is the finite cell line and second one is the continuous cell line. Let's discuss about the first finite cell line. This cell line which go through a limited number of cell division having a limited lifespan are known as finite cell line. These cells pass several times and then lose their ability to proliferate, which is genetically determined, even known as census. Cell line derived from primary culture or normal cells are finite cell lines. You can see in this picture also. This is the tissue samples and after trypsinization and it is start culturing and formation of primary cell culture is start here. This is the monolayer and then monolayer again convert into the different type of cells, but the number of cells are very specific here. Primary cell line is growing in this picture, secondary cell line is here, then tertiary cell line is also here, but cell division continuously up to finite division. But you can see in this picture the cell number of the cell are very limited here. As we discuss about the continuous cell line, then when a finite cells line undergoes transformations and acquire the ability to divide in indefinite, it become a continuous cell line. Cell culture are prepared in this way can be subcultured and grown, identified as permanent cell line. These cells are less adherent. These cells are less adherent. You can see in this picture, this is the again starting by the trypsinization method, and enzymatically we use enzyme digestions and mince or chop this process, and culturing is start here. This is the um, uh, tissue are growing. Tissue cells are growing here in this picture, and then primary cell line, you can see the number of cells are very higher as compared to this one, and then in the secondary cell line the numbers cells are again is higher as compared to this finite cells. So these cells are less adherent, fast growing. You can see here these cells are less adherent, fast growing, less fastidious in their nutrients requirement, able to grow up to higher cell density and different in phenotypes from the original tissue. Such cells grow more in suspensions. They also have a tendency to grow on top of each other in multi-layer or cultured vessel surface. You can see here. So, examples of the continuous cell lines: cancer cells produce infinite or continuous cell line. Properties of finite and continuous cell line is, you can see in this picture, this transformation is normal in finite cells, but in the continuous cells is the immoral growth and control altered, and the tumorigenics. And anchorage dependent, yes, this is anchor independent, but continuous cells is not. You can see by this picture the different type of properties of both cells. Serum requirement in the finite cells is very high, but in the continuous cells serum required very low. And if we discuss about the growth rate of finite cells is slow, almost 24 to 96 hours, but in the continuous cells the growth rate is very high, almost 12 to 24 hours. And if we are talking about the yield is low in the finite cells, but yielding is high in the continuous cells. And control parameter is generations time, tissue specific marker, but in this continuous cells these 10 characteristics. Now, process of animal cell culture. The process of animal cell culture includes the following steps. First one is the tissue explants. Second one is the cell extractions. Third one is the culturing in a nutritious medium, and last one is the subculturing. You can see in this picture, firstly in harvested cells and isolated cells with the use of appropriate enzyme. And in this picture, after this isolate cell, apply the isolate cells on to an appropriate growth media in a culture dish. This is the culture dish. and then culture cells by placing the culture dish in a cell incubator, and subculture cells to obtain a pure culture or to bypass some problems such as senses, and then directly verify the culture cells are of the cells type of interaction, and then last one is cells are ready to be manipulated or modified for experimental procedure. So let's start with some discussing about the tissue explant. What is the tissue explant process? The removal of tissue from the organs referred to as tissue explant. And if we talk about the cell extractions, then the cells can extract from the tissue either mechanically or enzymatically. The extractions is mostly carried out by the enzyme action or by the process of trypsinization. And now we are discussing about the culturing in a nutrient medium. After that, the cell is cultured either on solid nutrition medium or liquid nutrition medium. In a solid nutrient medium, the primary cells form a monolayer, whereas in liquid medium, primary cells appear as a cell suspension. Now, last we will discuss about the subculturing. It also refers to a passage of the cells. After the formation of primary cell, the subculturing is carried out that is important to continuously study or to grow the cell. This is the most important step in cell culture which help us to understand the cell type. So, we almost discussed about the process of animal cell culture. Let's discuss about the advantage of animal cell culture. First advantage of animal cell culture is animal cells culture make the use of a low amount of reagents. Second one is serial passaging maintain the homogeneity of the cell type. And next one is they provide controlled physiological condition, and next advantage is provide controlled physiochemical condition like temperature, oxygen concentration and pH. Disadvantage of animal cell culture is animal cell culture require high technical skill to interrupt and to regulate the animal cell culturing. It is a very expensive method to carry it out, and next one is the 10 times more expensive for same quantity of animal tissues. Therefore, reason for its use should be compelling. And now we will discuss about the applications of the cell line. In the different area, cell line is useful like vaccine production, toxicity testing, cancer research, gene therapy and immunological study. Let's start discuss about the vaccine production. One of the most important uses of cell culture is in research and production of vaccine. The ability to grow large amount of virus in cell culture eventually lead to the creation of the polio vaccine, and cells are still used today on a large scale to produce vaccines for many other diseases like rabies, chickenpox and hepatitis B. Application in toxicity testing is culture cells are widely used along or in conjunction with animal tests to study the effects of new drugs, cosmetics and chemicals on the survival and growth in a wide variety of the cell types. Especially important are liver and kidney derived cell culture. In the cancer research application is normal cells can be transformed into cancer cells by method including radiations, chemicals and viruses. These cells can then be used to study cancer more closely and to test potential new treatment. Next one is the gene therapy. Cells having a function Cells having a functional gene can be replaced to cells which are having non-functional gene and for which the cell culture techniques is used. And last application is the immunological study. Cell culture techniques are used to know the working of various immune cell, cytokine, lymphoid cells and interaction between disease causing agent and the host cell. So I hope you all be able to understand about the animal cell culture, its types and applications. If you have any query, any related to this, you can ask me by mail or by comments. You can follow us on the Insta, Facebook, Twitter, WhatsApp, YouTube. So thank you guys for listening me. Thanks again.